Okay, so I'm going to be presenting on the paper I wrote with Carina, Curto, and Igor Rivin on Betti curves of symmetric rank 1 matrices. Oh, yeah. So kind of the setup for this problem is just this fact that measurements in biology are often proxies for the actual variable we're interested in measuring. So an example in neuroscience is going to be calcium imaging. So how does calcium imaging work? With genetic mutation, recorded neurons increase with brightness when there's an increase in neural activity, an influx of calcium ions. So actually I have a video of this, but maybe it doesn't, it plays. Maybe.
No, I don't think
okay.
It's working.
But there's an increase of activity, and what you would see is that in this video that's not here is that when there are spikes in the electrodes, there's going to be an increase of brightness in this signal, but the signal essentially looks like a convolution with an exponential with the actual calcium signal. And so there's actually a nonlinear relationship between the neural activity and the recorded signal, but at least maybe it's a monotone. But why is this a problem? The problem is when you're going to analyze correlation matrices that's induced by this data that has a nonlinear relationship. The problem comes is that if you try to use linear algebra techniques to analyze this data, you know, a lot of these notions like rank or spectra, they're really, they're not numerically stable when you apply monotone nonlinearities. So in this example, right, I have a cartoon of a 100 by 100 rank 5 matrix. If I apply this nonlinearity f, this exponential nonlinearity f to every entry of A, and I produce B, well, if I look at the singular value plots, the singular values of A is exactly of a rank 5 matrix. There's only 5 non-zero singular values, but when you apply the nonlinearity, then it becomes a full rank matrix. So I guess then the question is, well, okay, so as long as we assume the data is good, that it's monotone, then the question is, well, can we find a matrix statistic that is invariant to monotone nonlinearities applied to entries? And maybe one way of doing this is going to be using topological data analysis. So again, we're applying monotone nonlinearity. So the only thing we can really guarantee is preserved is the ordering of entries in our symmetric matrix. So this technique, which is introduced in by Giusti and Carina in PNAS in 2015, it works as this. So in this example, we're going to take M, which is a rank 4 matrix, symmetric matrix, and I'm just going to order the entries. So for ease, I just, you know, I write it out. And we're going to ignore the diagonal. So now I have pointer. Okay. So we know that the smallest entry of M is going to be at this 4, 2 index. And so we're going to view this matrix M as the weighted adjacency matrix of a complete graph. And we're just going to add edges in order of their weight. So at this 2, 4 entry, 4, 2 entry is the smallest. So we're going to add an edge between nodes 2 and 4 at time 1. Next is going to be at 1, 2, and we're going to add an edge between 1, 2. We proceed in this manner, and we're going to create a sequence of graphs. And by filling in clicks, we're going to create a sequence of filtration of click complexes. Now, once you have this filtration of click complexes, you can start computing the Betti numbers of each one of these click complexes. And the Betti numbers of the click complexes in order are going to produce the Betti curves. So we see at time 4, when we've added in the fourth edge between 1 and 5, well, we create a hole, a non-trivial hole, because we can put one simplex here, and so we can record it. And just to note, right, again, any matrix with this ordering of entries will produce this same Betti curve. So it's an invariant to monotone nonlinearities upon entries. So once we have this structure, not only can you use it, you want to know, what theorems can you prove about it? So one of the theorems we're proving today are going to be about Betti curves of rank 1 matrices. Rank 1 matrices, and you can apply any monotone nonlinearity to it. So they have characteristic behaviors, and but we basically divide them into 4 different cases. And so quickly, we're going to just say a rank 1 matrix is generated by a row vector if it's, is X, M is X transpose times X. And then there are 4 cases. So if X is all positive, all the entries are positive, then we're going to call it positive rank 1. If all the entries of X is positive and we say M is minus X transpose X, we're going to call it negative rank 1. If X has negative and positive entries and M is X transpose X, then it's positive mixed signs. And X has negative positive entries, and it's minus X transpose X, it's negative mixed signs. Okay, so the first theorem is going to be the case of positive and negative rank 1 matrices. If we just look here, besides Betti 0, this is just counting the number of connected components of each of our clique complexes is decreasing because eventually we make a complete graph. We see that the Betti 1 and up, just in these computational experiments, they're all identically 0. So that's the first theorem. Let M be a positive or negative rank 1 matrix, then beta kt, the Betti curve, the kth Betti curve is identically 0 for all k greater than 1. So, So how does the proof of this work? Well, let's just think about a rank 1 matrix and its generating vector. We can just assume that it's written in linear order because, whatever permutation you have to apply to get it into linear order, that's just going to act on the vertices. So you just always have the same graph. Okay, well, we notice that if we have an edge, let's say we have this edge 2, 3, just by a simple inequality, if we have an edge between 2 and 3, we must have an edge between 1 and 2 and 1 and 3. Well, what does this do for us? Well, it means that actually the smallest node, the smallest vertex, is a coning vertex. And so in the positive case, first, each one of these clique complexes is a union of a cone or a sequence of isolated vertices. Well, but cones are contractible, and so it's going to be a homotopy equivalent to a set of points, and that's why we have the trivial Betti numbers. The same proof applies also in the negative rank 1 case. Why? Well, now our smallest entry is going to be just the biggest number, and so so now our coning vertex would just be 4. And so in the positive and negative rank 1 case, we'll have identically 0 Betti curves. Okay, so next are the kind of the two are the positive mixed signs and the negative mixed signs case. In the positive mixed signs, we see that the Betti 1 is going to be increasing linearly until around 0.5 or so, and then it drops significantly and it becomes all 0. But for Betti 2 and higher, they're all identically 0. In the negative mixed signs case, we're going to just have all identically 0 Betti curves. So how do we prove this? This observation we prove this we make an observation. Okay, so in the positive, okay, so we're going to take this mixed signs vector. Again, we can just linearly order them. Okay, now it's going to create a matrix. We generate a matrix, and it has this block structure where there's a negative part and positive parts. Okay, so in the positive case, right, we're adding edges from lowest weight to highest weight. So we're going to add these negative parts first. Okay, but this is essentially going to correspond to a bipartite graph. And then after this, we're going to add edges. Right, these, if we just restrict to, let's say, generated by, -3, -2, -1, and 1, 2, 3, 4, well, those will just be positive rank 1 matrices. So these are positive rank 1 matrices. We're adding cones. And so in the negative case, we're just going in reverse order, where we create these 2 positive rank 1 cones first, contractible simplices, And then we're going to start adding edges. But again, we're going to add edges in order of a negative rank 1 matrix. So that's kind of the key observation to prove those two theorems. So, okay, now we're going to get to the positive mixed signs case. In the positive mixed signs case, then the theorem is that for Betti 2 and up, it's all 0, and we have the bound. To prove it, essentially, we define this thing called the join. Well, I didn't, Milner did. And we have the join of a vertex, but essentially all a join in the simplicial case means is that if you have two simplices, but every node is connected, essentially there's a bipartite graph between all the vertices, well, then the homology is given by this. And by Milner, if we have A and B are acyclic, if the Betti numbers are all 0, and we know already from the positive case that on either side of the bipartite graph, those are acyclic, then the join of those homology is all 0. So that's essentially the proof of that. I should maybe write proof sketch. And that's how that works. Okay, so in the negative mixed signs case, it's a little more technical, so I'm just going to give more of an overview. Maybe I'll do it with this picture. So in the negative mixed signs case, right, we're going to build these two complexes first. We're writing them in this red part and this blue part. Okay, these already from the positive rank 1 theorem, it's already going to have 0 Betti numbers. Now, we start adding green edges, okay? So the green edges correspond also to positive or negative rank 1 matrix. We're adding edges in order of a positive rank 1 matrix. So let's just say we add these first 3 edges, okay? Here's the trick. We're going to say one simplex is going to be the union of this 2 is contractible. We're going to say another complex is these 3 edges union the blue part. That's contractible. We also know the intersection is contractible. So this is clear application of Meyer-Viator sequence, and then you get that the entire simplex or complex is contractible. And that will prove the negative mixed signs case. So there's a summary of the theorems. And so now we're going to applying it to calcium imaging data. With our collaborators at ENS, they've been collecting data using this very fancy microscope and collecting data from the Zebrafish optic tectome. It's this part here. It's primarily inhibitory cells. These zebrafish, they're like 10 days old. The nice thing about them is that these larvae, if you breed them properly, their skin is clear. So you can record 3,000, for this data, 3,000, 4,000 neurons simultaneously using the calcium imaging signal. We're going to take this data, we're going to deconvolve it with Witten's method. Is not the physicist, is his daughter. And we produced the correlation matrix, and we're going to look at the Betti curves induced by assemblies. The assemblies, maybe you can, I draw a picture of the assemblies here. Assemblies are groups of highly correlated neurons that are thought to pertain to function. If we look, so these are pictures of the assemblies. Now, if we look at the Betti curves induced by the submatrices of the correlation matrix induced by the assembly neurons, we see in these two cases, but it's very typical, for all of the assemblies, is you're going to get identically 0 Betti curves. So these are the rank 1 Betti curves, right, of the theorem. In comparison, I just pulling a random assembly. I'm just going to create some random neurons and of the same size, and I'm going to produce the Betti curves of that from the correlation matrix. And we see they're clearly different. This is more of a random structure. And so they're clearly different. However, we're gonna, and this is these are the Betti curves of a rank 1 matrix we proved. So in comparison though, with these submatrices, I have the singular value plots for all 4 of these matrices. They look basically the same. So it would be hard to detect the difference in dimensionality just from looking at the matrices. And so I guess endpoint here is that the topological data analysis lets us find structure in the correlation matrices of the neural assemblies that perhaps linear algebra cannot. And so I guess as I end, kind of interpreting this from the neuroscience perspective, right? The previous theorems of the identically zero Betti curves are evidence for kind of this lower dimensionality of the correlations or maybe the structure of the assemblies. And this is kind of in line with the way assemblies are thought to work in the brain, right? Assemblies are thought to be like specialists. They perform specific tasks. And these specific tasks are, a lower dimensional thing. So I guess Betti curves allow us to support this hypothesis. And so thank you. And any questions?
There is one question. Can you come here to get the question on Zoom? Because we cannot hear from there.
This is actually an elementary observation, but I want to figure it.
Yeah, here is good.
Okay.
Just speak clear.
Okay. It's just an elementary observation. Like when you construct the Betti curves, you actually use a particular ordering or the elements of the matrices. Now you will take 1, 2, 1, 3, 1, 4, and then you go to 2 something. If you take, for example, 1, 2, 1, 3, and 2, 3, you get a different Betti curve. And what I would like to understand is that because you get the whole at the third step. That's
okay.
What
yes.
I mean. Okay. And like, there is any specific reason to choose that ordering, or did you consider the fact that maybe changing the ordering, things will perform in a different way, or did you?
So are you talking about this example? In this example, I just went in Python and I just generated a random one.
Yeah.
And then I copied it down. I think that's kind of the nice thing is, well, you can have different orderings and still produce the same Betti curve, no, I didn't make this particular ordering for a particular reason.
Oh, okay. So maybe
just because I wanted to show the whole.
Okay. So maybe you could consider like as an additional parameter of bearing the structures, changing the ordering.
Oh, okay. So actually, if you do, let's say you permute the entries of this, essentially that's going to just correspond to like Erdo-Shreni graph.
Okay.
Right, and these have these has been studied by Matthew Kale, and they essentially Betti curves of random matrices look kind of like this. Okay. So actually, the ordering of the entries matter, right?
Okay.